Hello students, in this video I am going to tell you about the sexual reproduction in flowering plant, and this is my second video for this chapter. And in this I am going to tell you in detail about pre-fertilization event. As in earlier video I explained you about the flower. So this is pre-fertilization event. As we know that in pre-fertilization event there is two steps. First is gametogenesis, that is formation of gametes, and second is transfer of gamete, that is gamete transfer. So very first, that is gametogenesis. So here we will see about the formation of male gametes, that is sporogenesis or microsporogenesis. Or simply called as sporogenesis. So in pre-fertilization event, so many things are responsible for this, like hormonal, meristematic, embryological, and structural. So how many things are responsible for pre-fertilization event? That is hormones, meristematic tissues, embryology, and structure. And these all occur prior to flowering. These all occur prior to flowering or initiation of flowering. So in this shoot apical meristem is going to convert into reproductive meristem, that is flower. So you have to remember these points, that hormonal, meristematic, embryological, and structural changes occur prior to flowering in plant, and which leads to the formation of shoot apical meristem into reproductive meristem. So see here, this is as I told you that in this video we will see about the microsporogenesis. So this is the uh, anther and filament, that is stamen, male reproductive part. And once you cut a section of young anther, you can see like this. So there are two types of cells. You can say microsporangial wall, which has four types of cells, and then sporogenous tissue. So microsporangial wall, what does it contain? It contains epidermis, endothelium, middle layers, and tapetum. And you have to remember that these three layers are protective in function, and this very layer, that is tapetum, it provides nutrition to the developing sporogenous tissue. Nutrition to what? Sporogenous tissue. So you can see here This is the structure. This is a tetrasporangiate anther, and you can say the outermost is epidermis, then 2 to 3 layer endothecium. This is vascular strand, and these are the sporogenous cells. This is the stomium, from where the uh, anther can dehisce, and these are the middle layers, and the innermost is the layer named as tapetum, which provide nutrition to the developing sporogenous tissue. So epidermis, it is a common covering layer of anther. Endothecium, it is hypodermal wall layer of microsporangium or pollen sac, and middle layer. So see this groove, stomium. Stomium, it is a shallow groove between the microsporangia of an anther lobe, and it is as I told you that it form the line of dehiscence. You can see here, these are the line of dehiscence. So it is formed by the stomium when we cut the section. Then the last one is the tapetum, and it is the innermost layer of the microsporangial wall, and it provides nourishment to the developing sporogenous tissue. There are many more about the there are many more points about the tapetum, like first, it provide nutrition to the sporogenous tissue. Nutrition to sporogenous tissue. Second thing, uh, it secretes hormones like IAA, indole acetic acid, IAA, indole acetic acid, and that are stored in pollen grain for their early growth. These are stored in pollen grain for their early growth. Then next, tapetum also secrete Ubish granules. What is Ubish granule? It provides most of the sporopollenin. It provides most of the sporopollenin and other material for exine part of the pollen grain. Sporopollenin, it is very much important for your examination point of view, and sporopollenin forms a exine, as pollen grain has two layers, that is intine and exine. Exine and intine, you can say exine is the outermost and intine is the innermost layer. So some points about the sporopollenin, that it is the outermost layer of pollen grain, and uh, it is made up of highly resistant biochemical, that's the name sporopollenin itself. What is this sporopollenin? It is the biochemical which is highly resistant, and that biochemical is related to the group of cutin and suberin. Cutin and suberin. And how it is formed? It is formed by the oxidative polymerization of carotenoids. I think all these terms you are knowing, what is carotenoids and cutin and suberin. So, and sporopollenin is one of the most resistant organic substance, most resistant organic substance. So point to be remember, one of the most organic substance, resistant organic substance. It is not affected by high temperature, strong acid or alkali. Do not affected by strong acid or uh, high temperature or any alkali. No enzyme is known to degrade it because of the presence of sporopollen in pollen grains of the earlier plants, or you can say past plants, are well preserved as microfossils. So this is very much important, sporopollen, and you have to remember this point because it has been asked in examination. So this is all about the sporopollen, that it form the outermost layer of the pollen grain, that is exine. Biochemically it is cutin and suberin. It is highly resistant. It is formed by the oxidative polymerization of carotenoids, and most resistant known organic substance, not affected by strong acid, high temperature or alkali, and no enzyme is known to degrade it. So in earlier plant or past plant, that's why they are well preserved as microfossil due to presence of this microfossil due to presence of this sporopollen. So come to the tapetum, as we were discussing about the tapetum only. So first point was the it provide nutrition to the sporogenous tissues. Second point, it also secretes hormone like IAA or indole acetic acid that are stored in pollen grain for their early growth. Next point is it provides pollen kit. So this is will be the fourth point. It provides pollen kit. Do you know what is pollen kit? Pollen kit, it is uh, yellow viscous uh, substance present over exine, yellow viscous substance present over exine. And what does what is the function of this pollen kit? It allows pollen grain to stick on body of insect. So it allows pollen grains to stick on body of insect. So can you tell me that on which type of flowers is it present? So it is present obviously in entomophilous flowers. It means that it is present in the flower which are pollinated by the insect. And the last but not the least, that tapetum provides compatibility and incompatibility proteins. Compatibility and incompatibility compatibility proteins. Do you know what does it mean? Compatibility and incompatibility protein. It means that uh, a flower is not having any filter, that the unwanted pollen grains will not come to any flower. So due to lack of this filter, pollen grain has the flower has this capability to reject such type of pollen, this is not their pollen. So this type of character is called as compatibility and incompatibility protein. We will discuss in detail when we will study about the self incompatibility uh, in flowering plant. So for here this is all about the tapetum.